So the order of the presentation is going to be: first it is Suman, second Varsha Sharma, third Deepak Suman, fourth Priyanka Jadhav, fifth Sonali Tiwari, sixth Sangeeta Kumari and Anita Yadav, seventh Shweta Sharma, eighth Sandeep Yadav, 9 Mosmi Meena, 10 Jayashree, 11 Anirudh Khandelwal, 12 Miriam. So this is the order of presentation. Okay.
Okay, ma'am.
Some of you just in the afternoon has emailed me their presentation, so I'll be downloading that. But if you want to present it from your end, that is absolutely fine. Okay. I asked you to give me the presentation just in case there is any network issue at your end, so that we can resolve it at that time by sharing it from our end. Okay.
Okay, ma'am.
Okay, so then I think we can start, right? Good afternoon, everyone. I, Dr. Vijay Chaudhary, welcome you all to International Conference on Women and Careers in STEM, Representation, Challenges and Opportunities, organized by MIT University, Rajasthan, in association with University of Rajasthan, Jaipur. We are starting the oral presentations and poster presentations by the participants. Before we begin the session 3, let me introduce the session chair, Dr. Ira Upadhyay. Dr. Ira Upadhyay is currently working as Associate Professor at MIT University, Rajasthan, Jaipur since November 2015. She acquired doctorate in botany with specialization in environmental sciences from Aligarh Muslim University, Aligarh. Currently, she is engaged with monitoring and modeling of air quality, data analysis, risk assessment, occupational safety, and health research. She has presented her research work in national and international level conferences, seminars, and workshops. She is a collaborator of Global Burden of Disease. Dr. Ira is a fellow and life member of several international reputed professional societies, including Royal Society of Chemistry, Indian Meteorological Society, South Asian Meteorological Association, Air and Waste Management Association, etc. She is on the editorial boards of several international peer-reviewed and refereed multidisciplinary journals. She had authored 2 books, several chapters, and chief editor of special issue of international journals. Dr. Ira has guided more than 25 UG and PG research projects and currently guiding 4 research scholars for PhD degree. 1 PhD has been awarded under her guidance. During last 4 years, she has contributed mainly on global issues related to health. She has published 5 book chapters and more than 46 research papers in peer-reviewed and refereed journals, including Nature, Nature Medicine, Nature Human Behavior, Lancet, Current Topics in Medicinal Chemistry, etc. Her overall contribution to research papers is of 2,229 impact factors and 7,967 citations. Dr. Ira is also working on some novel concepts related to air pollution and herbal medicine and filed 4 patents, out of which 3 got published. With this short introduction, let me request our session chair to evaluate the participants. Most welcome, Dr. Ira Upadhyay, ma'am. Thank you. So participants, I think we can start with the presentations. First is Suman Prajapat.
Yes, ma'am.
Please share your presentation, Suman.
Okay. Ma'am, presentation is visible?
It's coming up. It's coming up. Yes, it's visible. Please put it on full screen mode.
Good afternoon to everyone. Today I am presenting presentation.
Suman, can I interrupt for you for a minute?
Yes, ma'am.
This is for everyone, all the participants. Would like to give you the instructions, repeat it once again. We have 7 to 10 slides, I believe, each one of you, and you have 5 minutes for your presentations, and 2 minutes will be for question answer session. So please stick to the time, everyone, because we need to finish it off within the time limit. Thank you. Thank you for your kind cooperation. Okay, Suman, please go ahead.
Good afternoon to everyone. Today I am presenting ethnomedicinal plants in adult sacred grove in Alwar, Rajasthan. Objective are ethnobotanical survey of selected group. Next is interaction with local community, and last one is study of ethnomedicinal plant species. Introduction. Sacred grove is the forest fragment of varying size which is protected by local community. The sacred groves are known as many names: church forest, fetish forest, and sacred forest. The picture is showing the relationship between sacred groves, ecology, and religion and anthropology. The next is methodology, and first is study area. Study area is adult sacred groves. The picture is showing this sacred site. Next is method. Ethnobotanical survey of selected sacred groves, interaction with local people, and ethnobotanical medicinal plants. The picture is showing the interview of local people, and next is meeting with women. Here is some important medicinal plant recorded from the sacred site. The common plant is Azadirachta indica, which is used in antiseptic as a skin disease, etc. These are also the other plant recorded from sacred site. These are some medicinal plant species of images. A is Electria aculis, B is Tridens procumens, C is Aderoda vacis. These are the other plant species which is found in sacred groves. Conclusion. From the study, 30 ethnomedicinal plant found in which is belonging to 70 genera and 15 family. These plants are used in fever, cough, cold, and skin disease. Study revealed that tribes living in around the sacred grove depend on the plant species for their health care. So we should conserve the sacred grove through the sustainable utilization of medicinal plant according to sacred grove. Thank you.
Thank you, Suman, for sticking to the time. Ma'am, you may ask questions.
So, Suman, what do you think? How can you conserve these plants? Is there any strategy in your mind?
Yes, ma'am. Ma'am, we can conserve plant through the worship around the worship of deities, we can grow plant around the temple.
Okay. Okay, thank you. Next group.
The next participant is Ms. Varsha Sharma. I don't see her here. Ms. Varsha Sharma. Okay, it seems like she's not joining. The next one is Deepak Suman. Ms. Suman, I think she's here. Yes. But it is Suman Prajapat. I don't see Deepak Suman. Okay, the next participant is Priyanka Yadav. Priyanka Jatav. Priyanka is here. Yes, Priyanka, please start your presentation.
हेलो
Yes, Priyanka, we can hear you.
एक्चुअली मैम मेरे फोन में ना कुछ प्रॉब्लम है, तो क्या आप स्क्रीन शेयर कर सकते हैं मैम हमारी पीपीटी की?
Yeah, I can do that.
Hello.
Hello. Priyanka, I'm opening your presentation. It is showing some repair. Let me see again. Just 1 second, Priyanka. Okay. Is it visible now? Hello, Priyanka. Can you see it now?
Yes, ma'am.
Okay, please go ahead.
Hello, everyone. I am Priyanka, and my presentation is the impact of anthropogenic disturbances on biodiversity and carbon storage in Sariska Reserve, Alwar, Rajasthan. And uh, mm, my objective in that topic, analyze the various levels of anthropogenic disturbances in the Sariska Tiger Reserve, and second is determine the impact of anthropogenic disturbances on the plant biodiversity in STR. And my study area in Sariska, which is located in Alwar, Rajasthan, and local people live there and dependent their biodiversity and livelihood. So many disturbances are there, and my part of sociological analysis is sampling site. I selected randomly, and on the basis of type of human activities and interference, and different region of core and buffer area. And my sampling conducted quadrate method, random quadrate method, and that size is 20 meter and 400 meter square shape. And that's are my pictures which are collected in my study areas. And this is my location area, which is located in Google Earth, and that are my data. Two sites on Beira and Raika, and that is मेरे anthropogenic disturbances parameters, and which are diversity indexes, Shannon diversity index, and Simpson. And these are my anthropogenic factors: lopping, cut stumps, grazing, regeneration status, and sapling on seedlings. And that's my result. And in two sites, Beira and Raika, the most dominant species in Beira site, that is 2.22, and Raika site species richness is 0.23. And that data shows that dominant species of site Beira is Anogenesis pendula and Vacella leucofolia, and Raika dominant species is Vacella leucofolia and Butea monsperma, Anogenesis pendula, and Holoptelia integrifolia. And that's are my references. And thank you so much, ma'am.
Okay, Priyanka, what kind of anthropogenic disturbances are there?
Ma'am, first cut stumps and lopping and grazing. That's are most of the anthropogenic disturbances, and some regeneration status saw there, and canopy cover.
Okay, so how this will affect conservation?
Sorry, ma'am, I don't listen. Otherwise,
How these will affect conservation?
ma'am, local people lives there and depended on her depend on her matlab, fuel food इनके लिए डिपेंडेंट है वहाँ के जो लोकल लोग हैं इसके वजह से वहाँ पे पॉजिटिव डिस्टर्बेंसेस थोड़े ज्यादा ही हो रहे हैं तो इसकी वजह से मतलब वहाँ का जो कम्युनिटी है वहाँ के जो स्पीसीज हैं वो विलुप्त होती जा रही है जिसकी वजह से मतलब हम वहाँ के लोगों को रिलोकेट करें तो शायद वहाँ का हम कंजर्व कर सकते हैं
Okay, so what's the reason of extincting? रीजन क्या है मतलब ठीक है इनका उनके ऊपर क्या फर्क पड़ेगा जो अगर एंथ्रोपोजेनिक डिस्टरबेंसेस हैं तो उनसे ऐसा क्या उनको हाउ कैन नॉट यू कंजर्व देम ऐसा क्या
मैम
इफेक्ट पड़ रहा है
प्लांट्स मैम आगे बायोडाइवर्सिटी क्या पूछ रहे हो आप
हाँ जी
मैम अगर लोकल पीपल वहाँ पे जैसे प्लांट्स का कट करेंगे ग्रेजिंग ज्यादा होगी तो मैम वहाँ के जो डाइवर्सिटी है वहाँ की जो स्पीशीज की रिचनेस है स्पीशीज मतलब जो मैनी स्पीशीज है वो धीरे धीरे मैम वहाँ कट ऑफ होने लगेंगी तो वहाँ
क्यों
की जो उस एरिया की जो बायोडाइवर्सिटी
क्यों
है वो
व्हाट्स
कम
द
हो
रीजन
जाएगी
यही मैं पूछ रही हूँ क्यों
मैम वहाँ के लोकल लोग उनको यूज करेंगे जिस वजह से और मैम
उनके ऊपर
जैसे वहाँ
तो उनके ऊपर क्यों डिपेंडेंट है वो स्पीशीज किस वजह से डिपेंडेंट है
मैम स्पीसीज
हाँ जी ओके एनीवेज थैंक यू
Thank you, Priyanka. So the next speaker for today is Sonali Tiwari. Ms. Sonali,
Yes, ma'am. Good afternoon, ma'am.
good afternoon. Will you be able to share your screen?
1 second, ma'am.
Yeah.
मैम आप ही शेयर कर दीजिए मैम
Okay, fine. Shall we
Can we start,
now?
ma'am?
Can you see the slide? I'm putting it in full screen. I think it is visible now. Yes,
Yes, ma'am.
Yes. Can please tell me when you want me to change the slide, okay?
Okay, ma'am.
Yeah.
Good afternoon. My name is Sonali Tiwari. I am here to present the topic of my research work, that is assessment of heavy metals contamination in terrestrial ecosystems of Jaipur, Rajasthan. Ma'am, please change the slide. That is introduction of my topic. My topic is heavy metals. So what is heavy metals? Heavy metal is the metallic chemical elements which have a high density and toxic very low amount. It is persistent in nature. Example of heavy metals can be zinc, manganese, lead, cadmium, chromium, etc. Carbon and nitrogen.
Sonali, there is some disturbance in your connection. Please keep it stable.
Okay, ma'am.
Yeah, now it is audible correctly.
Okay, ma'am. Ma'am, please change the slide. Okay. This is my study area. My study area is Jaipur. cultivated and forest land of Jaipur city. Climate of Jaipur is semi-arid and continental type. Soil is poor, humus content, and water holding capacity is low. Jaipur is flanked by river forests in north and east of the city. Sabi and Banganga is the mainly river which is close to the city. Drainage is mainly two types, that is Amanisha Nala and Mansagli. Vegetation is mainly scrub forest. Major kharif crop which is grown in Jaipur is groundnut and bajra and kharif pulses, while Rabi season की crops हैं wheat, mustard, barley. Vegetables mainly जो जयपुर में grow की जाती है, that is tomato, pea, chili, brinjal, cabbage. Ma'am, please change the slide. This is my GPA allocation where I will study. Next slide showing is my sampling size. These are my objectives. First is estimate the seasonal variation in heavy metals concentration in soil across different land uses. Next is study of heavy metal on soil enzymes involved in carbon, nitrogen, phosphorus, and oxygen reduction process. Next is to assess the metal contamination in soil across different land use using geochemical approaches, that means enrichment factor and geoaccumulation index. Then change the slide. This is my methodology. For the heavy metal assessment, we use the preparation digestion analysis of heavy metal by the AAS, by the diacid treatment, while physiochemical analysis of soil can be done by the routine methods like soil moisture by gravimetry, soil organic carbon by volcanic black metal, water holding capacity by percolation method, available phosphorus by Olsen method, and pH and electric conductivity by pH meter and electric conductivity meter respectively. Then please change the slide. This is the result of my pre-monsoon study. My study is seasonally, so this is my pre-monsoon study analysis. And while next slide is showing is my monsoon study analysis of physical analysis of soil sample. Then change the slide. Please change the slide.
You want the previous slide?
Study of effect of these two slides are showing about my results of pre-monsoon and monsoon season of physiochemical analysis of soil samples. That is determined by the enzyme will be measured by Thalmate method. Activity of acid and alkaline phosphatase will be determined by Tabatabai and Bremner method, by Vishnu by Iwaji and Tabatabai method. Data analysis will be done by statistical methods. That is my work. Significance of this work is by excessive amount of heavy metal damage the soil structure, composition, fertility. So I detect and assess the presence of heavy metal in soil. So its contamination in soil, extent of metal pollution, and this can be determined by scientifically. By researching, we can meet a standard quantity, so which cannot influence the soil quality and soil health, which discharged by industry to wastewater and their effluent. Heavy metal enter the ecosystem and harm the animals as well as food chain humans. By estimation, we can set the limit and stop the metal to influence the ecosystem and food chain. So, ma'am, by the study of my research, or during the study of my research, we estimate the heavy metal content of that area. So हम सब चीजों को estimate करके पता लगा सकते हैं कितना heavy metal soil में present है और वो किस तरीके से वो किस तरीके से human animals. So that is.
Okay, Sonali. So Sonali, can you name the enzymes on which you are working? Soil enzymes?
Hello, Sonali, are you there?
Yes.
Yeah, please answer ma'am's question, Sonali.
Sonali, can you please name the enzymes on which you are working?
Ma'am, dehydrogenase, beta glucosidase, and phosphomonosaccharide, acid and alkaline दोनों.
Okay, you didn't discuss about the impact on enzymes. So is there any impact?
मैम एक्चुअली अभी रिजल्ट्स मेरे पास अभी आए नहीं है पूरे से, इसलिए मैंने शेयर नहीं करूंगी.
Okay, so and then only I think we can guess what is the, I mean, you can conclude your study, right? This is not like just impact, just you have seen the heavy metals, right?
हाँ
Not the
मतलब अभी
impact
मैम
on enzymes.
जी मैम कॉन्क्लूज अभी
This is,
रेडियो.
yeah, this is incomplete actually, as per your title.
जी मैम.
Yeah. Okay. Okay then. Thank you.
थैंक यू मैम.
Next participant is Sangeeta Kumari and Anita Yadav. Any one of them here? Sangeeta Kumari or Anita Yadav? Hello. I don't know who is Shiomi, so I'm asking again. Sangeeta Kumari and Anita Yadav. Any of them here? Isolation of endophytic bacteria from Caparis decidua. Okay. Then the next participant is Shweta Sharma. Shweta Sharma here. Shweta Sharma. Shweta, are you here? No. Sandeep Yadav. Mosmi Meena. Sandeep will not be able to present because he is outside, I guess. Isn't it, Sandeep? He's not able to present right now. Hello? Yes, Mosmi. Mosmi Meena, are you here? Mosmi? No. Jayashree? Jayashree, are you here? Jayashree? Jayashree? Okay. Anirudh? Anirudh Khandelwal? Anirudh? May I know who has joined in the name Xiaomi? Anirudh Khandelwal? Okay, no. Anirudh? No. Okay, next participant is then Miriam Baybot. Miriam?
आपका ना शेयर ऑडियो वाला ऑप्शन होगा आपके में
अब आ रहा है?
आपका ना
Masood sir. Hello. Hello, Masood sir. IT person. Hello.
मैंने तो कर रखा है।
Hello. Hello.
Violence, the annual rainfall is below 500 millimeters.
हेलो
कहाँ रिपीट करूँ
इसमें शेयर साउंड एक बार स्क्रॉल करके हाँ अभी शेयर कर
Hello everyone, I'm very pleased to be here today to attend this conference online. My name is Miriam Baybut. I will share my research topic.
मुझे कौन है मुझे तो आपको आ रही है मुझे भी आ रही है मैं बोल रही हूँ आ रही है इसको चलने दीजिए आवाज नहीं आ रही उसकी आवाज हमारे पास नहीं अभी भी आ रही रिकॉर्डिंग हो रही हम बार बार ये करते हैं
It's of the genus Prosopis, and yet it is one of the only 3 species growing in in Asia, northern India, Persia, and the Arabian Peninsula. This is a distribution map of the tree, and this is the native presumed range of the tree because we could not sample over all the covered dashed zone, but based on the World Dryland Forest Map and as per what we have found in the literature survey, this is the native presumed range of Prosopis cineraria, and you can see it covers a huge part of the forested dryland area. Here in green, you can see what we call dense forest, so dryland dense forest, and here in brown, other wooded lands such as open forests. So Prosopis cineraria covers both, either dense forest or open forest, but it grows really in an arid, systems, in arid systems. So this species is a matrix species. It means it is not a rare species there, but it is typical from there, and it grows in Southern and Western Asia. It belongs to the Fabaceae family. It means it's a leguminous species having root nodules to fix nitrogen, and it is a very well adapted tree to desert condition with foliar absorption of water, and it is also what we call a phreatophyte. It means it has a very long root to get the deep water from the soil. These are some parts of the tree. So bark is here, very cracked bark. Here are the leaves, which are composed of leaflets. Here the flowers, which are growing in spike. Here are a bark with thorns. Actually, it's a very thorny tree. And here are the pods, green pods, and the seeds. These are some pictures of young sapling sprouting from the sand, and you can see this is a tree which is indeed very well adapted to desert condition. And because a picture is worth 1,000 words, this is a picture of a small cluster of Prosopis cinerea trees growing in the UAE desert that was photographed by Felix Hinden, a photographer. And here we can understand the magnificence of this tree in its full desert habitat. If you want to know more about ecosystem services provided by Prosopis cinerea, I invite you to read my paper, which is ecosystem services provided by P. cinerea in the dryland of Southern and Western Asia, which was published in Botany Letters in 2021. And there we will explain deeply about how the tree participates in each of the 4 components of ecosystem services in an ecosystem services approach. So to know, what we call ecosystem services are actually 4 types of services divided as per classification made by the Millennium Ecosystem Assessment Program in 2005, sorry. And we have on this figure, it is shown, it is well summarized. So here in the center are the supporting services, which represent the factors allowing successful functioning of the ecosystem, such as nutrient cycling. It means that when these basic properties are disturbed, an ecosystem cannot function properly. Then you have the provisioning services, which describe the material or energy output people obtain from ecosystems, such as, for instance, fuel wood. You have the cultural services, which are the non-material benefits people obtain from ecosystem through spiritual enrichment, cognitive development, etc. And the regulating services, which represent the benefit people get from the regulation of ecosystem, such as water pollution removal, for instance. This is a figure which will show how the tree is implicated in all the type of ecosystem services. So here in orange, plain frames, they represent the 3 main compartments of the ecosystem. The dashed colored frames represent the 4 categories of services. So in blue are supporting services, in golden provisioning services, in green regulating services, and in purple cultural services. Then you have white filled central boxes, which represent the services involving P. cinerea, and the green filled boxes with capital letters show active component of each compartment. Blue plain arrows will show primary consequences on supporting services, while the dashed blue arrow secondary consequences on supporting services. And the golden plain arrows will show primary consequences on provisioning services, and dashed golden arrows secondary consequences on provisioning services. Of course, a plus will show positive effect and a minus will show negative effect. Let us now get deeper into the main topic of today, which is the investigation of intraspecific genetic structure of Prosopis cinerea in Rajasthan. Prosopis cinerea is a foundation species and the emblem tree of the largest Indian state, which is Rajasthan. Again, I refer you to the first paper had published about ecosystem services and the importance of the tree in its ecosystem. What happens is a worrying decline in Prosopis cineraria's population density over the past few decades had been reported in Rajasthan. And genetic variation and diversity in populations are prerequisites for species to adapt to environmental changes. The objective of the study was to estimate the genetic variability of Prosopis cinerea for the whole of Rajasthan, with markers already developed for the species. We had a first hypothesis, which is that Prosopis cinerea would be locally structured to a finer geographic scale than that of Rajasthan. And a second hypothesis, which is that Prosopis cinerea would be structured as per the agroclimatic zone division in Rajasthan, which we will refer as AGZ division. So the sampling campaign was conducted in Rajasthan, the largest state of India. And in this state, 47%, actually this state contains 47% of all drylands of India. It means it's a very arid state. As I was telling, there is an agroclimatic zone division as per physiographical features as well as climatic conditions. So 10 agroclimatic zones were determined in Rajasthan according to rainfall, temperature, altitude, latitude, natural vegetation, and soil type and agricultural use of soils. So we have 5 regions which are divided into agroclimatic zones. So you have also, like if you want to know more about physiographical features, you have the western Thar Desert with barren hills, rocky plains and sandy plains, the Aravalli range running southwest to northeast starting from Gujarat and ending in Delhi, the eastern plains with rich alluvial soils, and the southeastern plateau, itself divided into 2 parts. These are some pictures of the field campaign, and you can see it fits in this program, in this conference theme also, sorry, because I'm a single female within all this team of males. So I think it's also appropriate to show this picture. So what we've done is we have collected fresh leaves from 414 individuals in between May and November 2016 in Rajasthan, India. And when the field condition allowed, trees were randomly selected along transects of 50 km long in the center of each agroclimatic zone defined by the survey of Rajasthan. Along each 50 km transect, we sampled 5 trees every 5 km, and we took care of taking these trees at a sufficient distance from each other to avoid the sampling of clones, because the tree can also reproduce asexually. For every sample collected, we took the GPS position. And when population density was high enough, we could sample 50 trees and less otherwise. For instance, in population 8, in the zone 8 and 9. We'll see it further. Out of the 10 existing zones, what we've done is we could collect trees within 9 of them. And leaves were collected on mature trees only, wrapped in aluminum foil, and then stored at -20°C before washing and grinding. And also to limit phenolic contamination, we took the sampling effort on the young. We put the sampling effort on the young.
Good evening, everyone. Now we are moving on to the offline presentation. So we are done with the online presentations. Maksud sir, we can call the session. Thank you everyone for your participation.